Hello, welcome to the first lecture of the subject called Basics of Molecular Modelings of Drugs, that is taught within the specialization Pharmaceutical Chemistry for the students of study program Pharmacy. The subject is taught in form of lectures and seminars, and as an e-learning support we use the Moodle course with the same name, where you can find all the topics of the lectures, as well the presentations from lectures, and also some instruction and training materials that will be used in seminars. And you can find there also the exam final test. As you remember from the basic course of pharmaceutical chemistry, we used the molecular models kits. From the history, the models developed somehow. They started with Radix models, or sort of the wire models. Later they were introduced Stuart Black or Corey Pauling Colton models that have the typical coloring, for example, red color for oxygen, blue color for nitrogen, and so on. An important breakthrough in the history in the modeling was the model of DNA that was constructed by Watson and Crick. Within this subject, we will be working only with virtual models, and from the history we know that the first virtual models were employed in 70s of the 20th century. Let's start a little bit more generally with the definition of computational chemistry. International Union of Pure and Applied Chemistry defines it as a discipline using mathematical methods for the calculation of molecular properties or for the simulation of molecular behavior. So it can include different activities such as synthesis planning, database searching, or combinatorial library manipulation. For us, very important term is computer-assisted drug design. Sometimes it's also called computer-aided drug design. So you should definitely know the abbreviation CADD. Eventually, UPAC defines also computer-assisted molecular design. So these methods involve all computer-assisted techniques used to discover, design, and optimize either biologically active compounds with a putative use as drug or compounds with desired structure and properties. The second definition is more generally. In the beginning, I would like to mention some historical milestones in computational chemistry. You do not need to know the precise dates or the years, but you should be aware at least about the centuries. So the history of computational chemistry is probably 100 years back. It started with Erwin Schrödinger, who developed the wave function that mathematically describes the distribution of an electron through space. The equation involves so-called energy operator H, so-called Hamiltonian. In 20s of the 20th century, Robert Mulliken developed a molecular orbital theory where electrons were assigned to orbitals across an entire molecule. 1 year later, John Lennard Jones introduced the linear combination of atomic orbital approximation for calculation of molecular orbitals. Again, 1 year later, Fritz Jones explained van der Waals forces as due to the interacting fluctuating dipole moments between the molecules. The wave function was also employed in the first accurate calculation of molecular orbital for hydrogen molecule by Mr. Coulson. In 50s, the foundation of conformational analysis was laid down by Derek Barton within the study of steroid chemistry, because in this period huge exploration on glucocorticoids started. In 1956, the first ab initio hard-3 Fock calculation on atomic molecules was performed, and in the 60s, first use of computer for force field calculation was published by Hendrickson. It was in study of ring conformations. This was the beginning of development of molecular mechanic calculations. 4 years later, Egon Wiberg employed Stiepy's descendant algorithm for geometry optimization. In the 60s, also the first drawing program for molecular structures was developed. It was called ORTEP, and it was introduced by Oak Ridge National Laboratory. So if you remember using molecular editor ChemDraw, which is part of the package ChemOffice. So the very beginning in this field started with the program ORTEP. It was definitely so much sophisticated as ChemDraw software. The history continued in the 70s with several discoveries or breakthroughs. Gaussian 70 was made freely available via the quantum chemistry program exchange. In the same year, the term computational chemistry was firstly used for description of a new scientific discipline. In 1973, the Cambridge Crystallographic Data Center made freely available 3D structure files with small molecules, and it is a widely used database until today. In the same year, Allengel described the model of hydrocarbons with a new force field, MM1, and the first molecular dynamic simulation was published by Warshall and Levitt. As you can see, some of the names of the discoverer or scientists are underscribed. It means they were awarded Nobel Prize for their discoveries. So you can see that quite many are depicted in this form, and these were really important discoveries in the history of computational chemistry. In 80s, Mr. Kunz and co-workers laid down the foundation for DOC algorithm for molecular docking, which was the starting point for a multitude of researchers of area of virtual screening. In 1985, GRID was published by Peter Goodford. It is a computational procedure for determination of energetically favorable binding sites on biological important macromolecules. In 80s, also the first automatic 3D structure generation program was developed, and very important thing, the first example of protein structure-based drug design was used for discovery of antiviral agent zanamivir. Nobel Prize was awarded several years later to John Pople and Walter Kohn for development of computational chemistry in quantum chemistry. And 10 years ago, the first millisecond simulation of a protein was performed using Anton, which is a computer designed specifically for high-speed molecular dynamics. 3 years ago, simulation of subatomic particles of lithium hydride and beryllium hydride was performed by IBM on a quantum computer with using a 7-qubit processor. In the same year, the Nobel Prize was awarded to Dubuche, Frank, and Henderson for development of cryo-electron microscopy for the high-resolution structure determination of biomolecules in solution. And in 2019, quantum supremacy used programmable superconducting processor, which was published in Nature by Mr. Arute and his team. I think we can mention generally that the computers in medicinal chemistry can be used for molecular and quantum mechanics calculation, for drawing chemical structures, for generating of 3D structures of molecules, for energy minimization, calculation of molecular properties, and also for conformational analysis. We can somehow divide computational chemistry into two parts. On the right side, you can see computational molecular design that involves several methods. With them, you will get more familiar during the semester in the following lectures. We can include, for example, comparative modeling, molecular modeling, virtual screening, pharmacophore modeling, molecular dynamic, and some other simulations. On the left side, you can see the field of chemoinformatics that includes chemical data integration, data curation and annotation, data analysis, and data visualization. There's something in common in the middle, so some of the methods together can include bioactivity profiling or predictive modeling as machine learning. We will start a little bit with the process of pharmaceutical companies, how they search for new drugs. So in the first step is drug discovery, then the second one is the drug design. We talk about rational drug design, and the last step is drug development. So let's have a look at the particular steps. The first one starts for the company with a strategic decision of choosing a disease that should be hit by potential drugs. Within the pathophysiology of the disease, it's necessary to choose a target and identify a bioassay that can be used for identification of potential drugs that can be employed in testing. It is called screening. Within this process of screening, where you can use different libraries of compounds that are available in pharmaceutical companies, you find so-called hits. It means compounds that have shown an biological activity in the bioassay. The second step, drug design, involves the process from hit to lead compound. It's practically application of all techniques leading to the discovery of new chemical entity with specific properties required for the intended application. This is the UPAC definition. And in this step, the hit molecule or molecules are somehow modified um, with the intention to explore structure activity relationships, identify the pharmacophore in the molecules, and also drug optimization is important in the terms of optimal pharmacokinetic properties. The last step in the whole process is drug development. It's practically getting drug or the candidate molecule to the market. So it employs the preclinical trials, clinical trials, and patenting regulatory affairs. Let's have a look at the process in a picture. So the discovery in molecules can happen within some real laboratory screening, or nowadays the computers are employed in virtual screening. The virtual screening is called in silico because silicon is used on the computer chips. So you should remember this. The hit molecules from virtual screening needs necessarily to be either synthesized in case they are available. They are directly tested in bioassays in libraries, firstly in vitro. If the molecules are promising, they can be tested in vivo in different animal models. And if they have plausible properties, they go to the clinical phase of testing on humans. On the right side, you can see the process of clinical testing. The last phase of clinical testing goes then to licensing, which means registration of the drug in the market as a trademark or registered mark. Later on, the phase 4 is the pharmacovigilance phase when the drug needs to be still observed and some side effects needs to be checked, and it's a matter of clinical pharmacy. So I would like to mention once again the methodologies for lead compound identifying. The pharma companies employ so-called high throughput screening, which is a massive screening of nature extracts, corporate compound collections, and combinatorial libraries. Before that, the virtual screening can proceed in silico. Usually, computational docking to 3D structure of the target is used, and also some computational filters can be employed. As far as the compound libraries are concerned, the screening collections of real compounds can have up to 10 to 6 compounds, whereas the virtual libraries are really larger. They can have 10 to 12, up to 10 to 18 individual compounds. The scientists calculated how big is actually the chemical space of drug-like molecules. by combination of specific elements and employing also some uh, specific requirements for drug-like molecules, and they define the space as 10 to 63 of drug-like compounds, which is a huge number. Here you can see the diagram or scheme. From the beginning, we try to hit the target. We use either fragment-based libraries or the extract, combinatorial and corporate libraries. We employ consequently also the virtual library screening in silico, and in case some hits are found or candidates, they need to undergo the real screening in laboratory. And after the optimization of ADME profile, you can get a lead compound. Let's focus more now on the subject and definition of molecular modeling. It's actually investigation of molecular structures and properties using computational chemistry and graphical visualization techniques in order to provide a plausible three-dimensional representation under a given set of circumstances. I think the computer-assisted molecular modeling is practically the same definition because the computers are used always today. Generally, the computations employed in the molecular modeling can be based on two approaches. The one is so-called molecular mechanics, MM, and the second one is the quantum mechanics, abbreviated QM. The quantum mechanics is based on the Born-Oppenheimer approximation, and it says that the mass difference between nuclei and electrons is sufficient so that their motion can be considered separately. Proton is 1836 times heavier than an electron. So the quantum mechanics calculations are focused on the electronic state of molecule given a fixed position for the nuclei. So the electrons and motion of electrons are important and calculated in QM approach. Whereas molecular mechanics calculations are much more simply, they are focused on nuclear coordinates of a molecule at its ground state. So the atoms are considered as whole spheres, and the calculations are much simpler. UPAC defines also these two methods or approaches. So as for the quantum mechanical methods, they can be split into two methods. The first one are ab initio, it means from the beginning. These are non-empirical quantum mechanical methods of quantum mechanical calculations dependent of an experiment other than the determination of fundamental contacts. The methods are based on the use of full Schrödinger equation to treat all the electrons of chemical system. In practice, approximations are necessary to restrict the complexity of the electronic wave function and to make its calculation possible. To make the calculations easier, to simplify the quantum mechanic calculations, the semi-empirical methods were employed, and they use practically parameters derived from experimental data to simplify the computations. I don't like too much into detail about the QM methods because we will not be concerned with them during the seminars, during the practical part of our classes. We will be employed more with molecular mechanics calculations, so-called force field calculations. These are methods intended to give estimates of structures and energies for conformations of molecules. The method is based on the assumption of natural bond lengths and angles, deviation from which leads to strain, and the existence of torsional interactions and interactive and or repulsive van der Waals and dipolar forces between non-bonded atoms. Just to compare these two approaches or these two types of calculations, molecular mechanic calculations are limited to molecules containing thousands of atoms. You can see the list of applications, whereas quantum mechanic approach, especially the ab initio calculations, are limited to molecules containing tens of atoms because the calculations are much more complicated. The semi-empirical approach enables calculations to molecules containing hundreds of atoms. You can see again the applications in the list below on the right side. Now I will show only 2 slides focused on quantum mechanics. The grounds were laid by Albert Einstein, who defined the wave-particle duality, and the wave function denotes the psi that describes properties of quantum mechanical system and has a value at every point in space. So I already said that in QM calculation, the localization of electron density can be computed, and there are different techniques, for example, the Hartree-Fock method, then the semi-empirical quantum mechanics calculations, and lately density functional theory is heavily used in some scientific works that directly calculates the electron density distribution and the total energy, electronic energy. This is a revision, the scheme on the left side of applications using the Schrödinger equation with the psi parameter, and you can see the equation on Erwin Schrödinger grave. It's probably in Austria because he was an Australian physicist. I mentioned that molecular mechanics is more important for us. This technique originated later within the vibrational spectroscopic community. And energy of the molecule is defined in terms of nuclear positions. It means that electrons are not studied, and we assume that they adopt an optimal distribution around the nuclei. The energy of molecule in molecular mechanics has contributions from bond stretches, angle stretches, out of the plane bending and torsional energies with additional terms to describe non-bonded terms for van der Waals forces and electrostatic interactions. So you can see it here in the picture. You may remember how the infrared spectra are measured and the molecular mechanics is connected with the community of spectroscopists. So the energy of the whole molecule is a contribution of energy resulted from bond stretching, angle bending, bond rotation, and also from electrostatic interactions and van der Waals interactions. So the terms in molecular mechanics are again repeated here. The bond stretching is based on the Hooke's law. You know it from physics, from angle stretching, torsional energies, and composite function uh, uh, comprising the energy resulted from van der Waals forces and from electrostatic forces. So the force field is defined by a combination of mathematical energy function and a set of required parameters. The first type for calculations, force field calculation was MM1, introduced by Allinger for hydrocarbons, and it is applied in drug discovery for conformational analysis and protein ligand interactions. The electronic distribution and electrostatic isopotential can be depicted also in the methods of molecular mechanics. It is based on some positive or negative centers in the molecules or dipole moments. Now we came to one of the specific methods of drug design. So we mentioned firstly the two approaches of for calculations, molecular mechanics, and QM, quantum mechanics. Within our subject, we will use only the molecular mechanics calculations, and as for the methods used for drug design, we can split them into structure-based drug design and ligand-based drug design. So the structure-based design is a design strategy for new chemical entities based on the three-dimensional structure of the target obtained by different methods. So in this design, we are aware or we know the 3D structure of the target macromolecule. The target macromolecule can be, for example, a receptor, enzyme, nucleic acid, either RNA or DNA, and so on. So it's necessary to know the 3D structure, not only the sequence of amino acids in case of proteins, but also the three-dimensional structure of the binding site within the protein. So it's the most familiar application of computational chemistry, and the very first structure-based drug design was published by Bedell in 1976, which was focused on allosteric binding to hemoglobin. How can the 3D structure of macromolecule can be determined at atomic level? So experimental methods are employed. The most important is the X-ray crystallography, the crystallographic analysis. It's a versatile method in case of globular proteins. The protein that you want to explore from the point of 3D structure must be firstly expressed in specific microorganism. It needs to be isolated, purified, characterized, and crystallized. So the crystal is a condition for X-ray crystallography, and there are specialists who focus on this protein crystallization and the X-ray experiments and analysis of the data obtained. The second most important method is nuclear magnetic resonance. Its advantage is the determination in solution phase. It can be applied to the proteins up to 30 kilodalton, and it's synergic with X-ray. Another method that is used for 3D structure determination for proteins is neutron diffraction. We mentioned in the historical overview in the beginning of the lecture also cryo-electron microscopy. These are all experimental methods. The last method, homology modeling, is a computational method that can be used for prediction of 3D structure of protein. It is based on comparing comparison of familiar proteins. If you know the 3D structure of a familiar protein that has a specific similarity in amino acid sequence, you can predict the 3D structure of an unknown protein. The 3D structure data are freely available at the website of Protein Data Bank. It is so-called PDB database. And in the right upper corner, you can see that it involves more than 169,000 macromolecular structures. So in the table below, you can see in columns the type of method that was used for the 3D structure determination. You can see that the X-ray is the mostly used in comparison with the NMR or electron microscopy or neutron diffraction. It's also possible to combine the methods and use some hybrid methods. In the rows, you can see the type of macromolecules. So the proteins are the mostly representatives, whereas nucleic acids or complex of protein and nucleic acids are less represented here in the table. So if you have suitable software where you can render the molecules, you can upload files from this database. You can see here a plot growth of molecule contributions in the PDB database. It started in 90s. The growth was firstly linear. Around the year 2000, it grew exponentially. Nowadays, the growth is again linear. Here is an overview of key databases from a lecture of Professor Kocha. We will have one lecture focused only on databases, so I will skip this slide. Just to remind you, the structure-based drug design is based on knowledge of 3D structure of the target of the macromolecule, and it is applied in order to discover leads in an existing collection of molecules or select a compound from possible molecules, virtual molecules, which could be synthesized as a part of a lead optimization program. There are several steps in the structure-based drug design process. One of them is the molecular modeling. That is a computational method that predicts the 3D structure of protein-ligand complex. So it's a method for generation of possible pairs of the ligand and protein. So it generates a pose that shows a ligand's orientation within the binding site. For evaluation of binding energy, the scoring is applied. It predicts the free energy of protein ligand binding, so the creation of the complex. Another method used within the structure-based drug design is the molecular dynamics. It is defined by UPAC as a simulation procedure consisting of computation of the motion of atoms in a molecule or of individual atoms or molecules in solid, liquids, and gases according to Newton's laws of motion, which means it's based on the molecular mechanic calculations. So it's actually simulation to predict the time-dependent behavior of the model or of the molecules. Generally, structure-based drug design is used during the process of hit identification within the high throughput virtual screening, then later in the process of hit-to-lead optimization, and also during the late-stage optimization. In this scheme, you can see how the molecular modeling is connected to different activities, for example, combinatorial chemistry, screening, protein biophysics, molecular cellular biology, genomics, and proteomics, animal models, toxicology, and in pharmacokinetics. All these activities are connected with molecular modeling. Now I would like to turn your attention to ligand-based drug design. So we discussed already the structure-based drug design, and now we will be employed with the ligand-based drug design. This approach is based on the fact that we do not know the 3D structure of the target. We only know the 3D structure of active compounds, so-called ligands. We can start the ligand-based drug design with three different approaches. The first one is the compound similarity search. The second one is the pharmacophore screening, and the last one, the third one, machine learning, so-called quasar models. The first approach is based on the assumption that similar compounds exhibit similar biological activity. The compounds can be compared in structural terms or in property terms. So as for the structural terms, the compounds are evaluated in numbers if they, for example, some features are contained in the molecule, for example, hydrophobic or aromatic moieties, ionizable groups. In terms of properties, we can describe molecules, for example, with calculated logarithm of partition coefficient, or with molecular weight, with number of rotatable bonds, or number of hydrogen bond acceptors or donors, or pK value, and so on. The second approach in ligand-based drug design is the pharmacophore screening. So the ground was laid by Emil Fischer with the Locke-Key hypothesis applied to drug and the target. The real definition of pharmacophore came with Paul Ehrlich, who defined the pharmacophore as a molecular framework that carries the essential features responsible for a drug's biological activity. However, Peter Gund modified the definition of pharmacophore as a set of structural features in a molecule that is recognized, or that are recognized at the receptor site and are responsible for the molecule's biological activity. So this is, in my opinion, the best definition of pharmacophore or pharmacophore model. So the question is where to get the 3D structures of small molecules, of the ligands, of drugs or active compounds. You can get them from the Cambridge structural database, or sometimes it's you can see the logo, it's abbreviated with CCDC. It means Cambridge Crystallographic Data Center. The files with the 3D structures are available at the given address, and you can see the number of the organic molecules contained in this database. In this year, it's more than 1 million of compounds or structures. This database started 20 years earlier than the protein data bank. There are also some other databases of organic compounds. For example, zinc database is freely available, and it contains commercially available compounds. There are also some other databases. There are specific companies focused on creation of databases or trading with the databases, either real databases or virtual databases. Very common feature of databases is the usage of smiles code. It's actually a simplified linear record of chemical structure. So you have a example of a phenol molecule here, and you can see the smiles code below, which is alphanumerical code, and you can go through the blue links to internet and see the elucidation that the SMILES code are simplified molecular input line entry system codes, and there are some specific rules for the recording of molecules or the structure of the molecules. So how the pharmacophore model can be generated? There are 3 steps. The first one is identifying of common binding elements that are responsible for the biological activity. Then the second one is generating potential conformations of active compounds. And the last one is the measuring distances between binding pharmacophore elements in each conformation. Here are examples of some softwares used for pharmacophore generation, and I would like just to mention typical or common pharmacophore features. These are the hydrogen bond acceptors or donor centers, positive or negative charges, ionizable centers, also the surface accessible hydrophobic regions like aliphatics, aromatic centers, non-specific features, and also the flexibility of the molecule. So here you can see an example. The starting 2 active molecules, this is a very small set of molecules, contains some specific features, and below you can see the pharmacophore model created from these 2 molecules. So on the left side there is a nitrogen employed in a ring, which means it's bound to different substituents. Then there is a carbonyl involved in the molecule and aromatic ring center. The distance between the centers, between the features is measured, and it is actually 3-dimensional position of the pharmacophore features. I'm sorry, the slide is in Czech, but I will elucidate. So in the starting set, we have compounds that showed some activity on a specific target. We can train this set of molecule, constructed as a database. We can generate 3D conformations, and based on the conformation, create a pharmacophore model. We can somehow work with the model and make it more precise. The model will look like that in this picture, and for prediction of active compounds, we usually use a database of compounds with the unknown activity towards the specific target, and we can do the pharmacophore screening to get the list from this library based on the pharmacophore adaptation. The last approach in ligand-based drug design are the quasar models that can identify biological activity on some equation calculations. You know the abbreviation quasar. It is quantitative structure activity relationships, and the first or the lowest level was based on a relationship between activity and physical chemical properties. You may remember the calculations from the basic course of pharmaceutical chemistry, where you try to calculate the lipophilicity of compounds from one therapeutic group, and then you compared it with the experimental data. There are some more sophisticated quasar models that include structural patterns or connectivity. 3D quasar employs also the electrostatic and steric properties in 3D. 4D quasar computes with more conformations of ligands. 5D quasar adds induced fit models, and 6D quasar adds also the solvation models. So these calculations approaches merge nowadays somehow with artificial intelligence and creation of neuron nets. I would like to mention also the fragment-based lead discovery. The first publication with this approach was appeared in 1996 by the pharmaceutical company Abbott. They used a fragment library for structure-based drug design, and I would like to mention what is such a fragment. It is a small molecule with low molecular weight, and the fragment can show some activity in either virtual screening with known 3D structure of the target, or in a bioassay. Fragment screening can provide choice in the number and diversity of chemical starting points, and it enables identifying novel scaffolds and binding motifs. So the size of fragments can differ in different literature sources. This one shows the molecular weight from 110 till 250, or it can be 190. You know that lead compounds have the molecular weight around around 300 or 400. So in this scheme, you can see the real fragment library or virtual library of fragments. From the screening, we can get some hits. The fragment need optimization. The effectivity or activity of the fragment can be supported by X-ray or NMR guided model, and we need the process to enlarge the molecule of the fragment. It can happen in with the different approaches. It can be either linking of the fragment with another active fragment through a linker, or the second approach is growing of the fragment, which means that we can somehow fill in the empty space in the cavity in the active site of the target, or we can merge different fragments together that can be fitted into the active site of the target. Here's an example of enlarging of the starting fragment into a compound that resulted uh, into an active molecule that came into clinical test. So you can read the legend for this scheme. Here is a process of selecting active compounds from a fragment library or compound library. Usually filters are employed. You can see the filter here in the scheme on the right. The filter involves specific molecular weight. In this case, the fragments were searched. The unwanted functionalities or molecules with unwanted functionality were excluded. And also the criterion concerning the solubility was employed. The next step, so the starting library containing nearly 2 million compounds was limited to a library with 43,000 of compounds. Then the pharmacophore model was used for the screening, and also some fingerprint searching was employed. It will be elucidated in another lecture. So the library was limited to 7,010 compounds that were manually inspected by medicinal chemist, and limited to 1,300. And then the last collection of the compounds was 1,200. That was worked and tested in laboratory. Actually, the filters generally can employ molecular weight, toxicity, and reactivity. There are some unwanted functionalities in molecules of drugs, for example, aldehyde groups, terminal alkenes, thiols, anhydrides, and so on, nitroso groups. Just to compare structure-based drug design and fragment-based lead discovery. Within the structure-based drug design, we screen relatively small libraries with thousands of molecules. And many compounds need to be synthesized, whereas in the fragment-based lead discovery, there is usually a choice in number and diversity of chemical starting points. The chemist has small start points. These give opportunities for developing highly efficient low molecular weight lead, and this approach enables identifying novel scaffolds and binding motifs. We should mention also the conformations in molecular modeling. The history started with conformation of cyclohexane and orientation of the bonds into equatorial or axial position of the ring. The preferred conformation can be determined through calculation of relative energies of accessible conformation, and the calculation proceeds in this way that each rotatable bond is systematically rotated via a fixed amount, whilst the bond lengths and angles are kept constant. The objective is to discover all accessible conformations, which will lie at energy minima. As you can see, with the number of rotatable bonds, the conformations grow exponentially. There are different algorithms employed in conformation analysis, and you can see that we want to try the global energy minimum, not only the local energy minimum. So methods for conformational analysis can involve systematic search procedures, serendipity methods, or molecular dynamics. You probably know this diagram, the dependence of energy on torsion angle of butane. You probably do not know the Ramachandran plot, which is a dependence of phi and psi angle in the scaffold of proteins. There are some preferred regions with specific values of the angles. It will be discussed also in some of the later or the following lectures. The pharmaceutical or medicinal chemists employed often molecules some conformational constraints, so restriction of bond rotations. For example, you can see the molecule of remoxyprid below on the left that has the methoxy group creating a hydrogen bond with the hydrogen of the amide bond. There are some specific linkers with concrete values of angle for binding that are used in drug design. In the end of the lecture, I would like to sum up the methods. So we can know the target, or we do not know the target of the ligand of the inhibitor. So in this case, we have to use the ligand-based drug design in case we know the active compounds. It's an advantage to know the 3D structure of the target. So you can work during the molecular modeling also with the macromolecule. In case we do not know any active compounds, but we know the 3D structure of the target, we can apply the approach that is called de novo design. So we can design a compound based on the knowledge of the 3D structure of the active binding site, which can in some cases bring non-synthesizable molecules. So this approach might be a little bit dangerous from this point of view. And not knowing target, not knowing compounds. It's sort of serendipity screening, I would say. Not much rational. So here is an comprising scheme, so you can study yourself and check all the methods that were mentioned in this lecture as homework. In the end, I would like to thank you for your very kind attention and show you some websites of institutions that are concerned with computational modeling in the Czech Republic. These are not only big pharma companies, but also the Elixir company, for example. There is a research center, CADD, in Brno, and also some research groups are familiar with the computational method. For example, the Institute of Organic Chemistry and Biochemistry of Academy of Sciences in the Czech Republic, as well the Palackého University in Olomouc. So thank you very much. If you would have any questions, you can contact me, or you can expect more in detail elucidation of some of the methods or terms during the following lectures. Thank you. Bye.